Okay, so what I'm going to do now is just leave this channel open for the next few minutes, and if you've got any last questions or anything like this that you want me to go through for the Unit 4s, then now is your chance to ask them. So any questions at all, feel free just to pop them in the chat window, and I'll do my best to get through them in our next few minutes. Come here. Who did? So if you've got any last questions about the B4C4 or P4 exam, then now's your chance to ask those questions. So if you post them in the chat window, then I'll do my best to get through them in the next few minutes. Odie. And I even brought Odin as the mascot for you. Sit. So you've got about 10 minutes. If there's any of the last things that you want me to go through, then post it in the chat window and I'll do my best to go through them for you. So any B4C4 or P4 questions, then post them up in the chat window and I'll do my best to go through those for you. Yes, it's Odin. I did promise I would bring him in for your video. Hold on. I'll tilt the camera down. There you go. So there is Odin for you, as promised as well. So just as a last little good luck mascot, and now he's going to leave for you. So if we've got any questions, then post them up rather than just looking at Odin, as much as I'm sure that you would all love a permanent webcam on him. then we're not going to spend all night here, I'm afraid, folks. So any other questions, anything that I missed that you want me to go through urgently tonight before I see you tomorrow, then now is the time to ask. You have 8 more minutes to ask any questions rather than just sitting there watching me, watching my screen, which is not a riveting way to spend our evenings. Another example of covalent bonding. Not a problem. Okay. I've already done water and carbon dioxide, so let's have a go at, let's do chlorine gas. So chlorine gas, Cl2, okay? Now, chlorine itself has 7 electrons in the outer shell. So when we're actually drawing it, we're going to have 2 chlorines. So you can draw the 2 chlorines themselves next to each other with their orbits already set up. Now, each chlorine has 7 electrons. So all we're going to do is start off by putting the crosses in one. And we know that one of them must be shared because obviously it's going to need 8 in total, which means 1 is going to be a shared electron. Do the same on the other one, just using the dots. So put in your 7 dots, making sure 1 is in the shared zone in the middle. So that's just your covalent bonding of your chlorine molecule then. So the key thing to remember is that once you've actually got the number of your electrons from the periodic table, it's just a simple case of making sure that to make the full outer shell, you've got the right number being shared. So virtually every case they're going to give you, there will be 8 in the outer shell. So work out how many they've got to share to give each of them 8 actual electrons in that outer shell. Okay. Obviously, if you want me to go through the covalent bonding again tomorrow in lessons, then we can do that. Particularly if you're in 1 and 2, I'll give you that time for us to then just go through any of those bits you wish to ask me particularly. And then 3 and 4, we'll do our usual kind of slow stroll through all of the unit 4s from start to finish. So anything other than covalent bonding then. So answer Molly's. I can see there's 6 of you here at the moment, so I'm not that interesting to sit and look at. If you have any other questions, please ask, and then we can go through those as well. How do you control nuclear fusion and fission? Okay, so in terms of controlling our reactions within our nuclear reactor, then I'm not going to even attempt to draw this diagram because it will just look terrible. But what we find is, as we know, in our reaction, then we're going to end up with 3 neutrons being released from each atom. So what we end up with is a reaction that can get very, very big, very fast. So if you imagine that you've got this kind of setup, so you start off with 1 atom, that releases 3 neutrons, they hit 3 atoms, they release 3 neutrons apiece. The way that we control this to stop it going out of control is by using these amazing things called control rods. There's a lot of controls in there, I know. But the control rods quite simply absorb the extra neutrons. So that in a nuclear reactor, what we actually do is that once the reaction is going fast enough, we lower the control rods down, they absorb the extra neutrons there, and that then means that we can keep the reaction happening at a safe rate. And that's obviously what didn't happen in Chernobyl out in Ukraine, because they had that obviously big accident going back to the 1980s where the control rods didn't work and therefore they went into the meltdown situation. In terms of whether you've got to know about how glucose is stored and converted, then yes, you will have to know about the fact that it does get changed into different things. So if you're actually thinking about glucose itself, then what we find is that we don't just store it as glucose because that just messes with the whole water balance within our cells. So instead, what we do is we convert the glucose into things like starch. So just join lots and lots of glucose together to make starch. and that doesn't affect the water balance. We'll also change glucose into things like cellulose to make cell walls. We'll change it into, obviously, proteins, fats, all of these other things, as well as using it in our respiration in order to generate energy. But you're not going to be expected to know huge amounts of information there. For the higher tier, you're more likely to be asked about why glucose is stored as starch. And all we need to say there is that it's insoluble, so it doesn't dissolve, and it doesn't affect the water concentration inside the cells. However, it can be converted back into glucose readily as soon as we need it for respiration. So that's pretty much all you're going to need to know in terms of your glucose, how we change it and how we store it. So has anyone got any other questions? So any other questions about any of the B4C4 or P4 that I can go through to help you out tonight before obviously you go and have a nice early night and contemplate everything we've gone through tonight? There's now 8 of you watching here. I'm sure some of you must have some questions. Thank you for all those hearts, Callum. Transpiration. Okay, so transpiration stream. What we start off with then is over here, this is our root, okay? So in the actual root of the plant, we've got the root hair cells, and the whole reason we've got root hair cells is to increase the surface area. So water moves into our actual root hair cell, first of all, just simply by osmosis. Then it travels through all the cells of the root, and then what we find is it goes up the xylem, because remember, water transported in the xylem, all the way up into the leaf, and there it's going to travel down through the spongy mesophyll. And we know it's a spongy mesophyll because it's got the big holes for the little air spaces, just like a sponge has all those holes in it. And it's going to evaporate through the stomata. So what we find there is, because the water is coming from the roots up the stem into the leaf and then evaporating, it's this constant stream. So it's like a chain of water that's just getting sucked up through the plant. And as it's being moved, it's also carrying all those different dissolved minerals as well. So transpiration itself is just the loss of water vapor from the leaves. And then the transpiration stream is the movement of the water from the roots up the xylem and then through the leaves and out. Okay. If we're asked to do a kite diagram, if in site 1, for example, there's 20% of an organism, do you halve it or do you do 20% on each side? Okay. What you'd actually do there is they would give you numbers, okay? But the distribution of your kite diagram should always be evenly split across the two sides. Ooh, and that diagram's gone slightly wrong. But what you'd find is that if you had, say, 10 organisms, you put 5 below and 5 above it. So always split them nice and evenly between the two sides of your central line to show the distribution properly on your kite diagram. They're more likely to actually give you a kite diagram and ask you to interpret it than to ask you to draw one, in all honesty. Right, radioactive dating in rocks. Okay, so in terms of the radioactive dating in rocks then, what we find is that this is relying on the old half-lives. So when we've got our rocks, then it's going to be made of the uranium atoms. Now, uranium, as we know, is unstable, which means it's going to experience radioactive decay. As those rocks then undergo the radioactive decay, and the uranium atoms are going to then be converted into lead atoms. So the actual rocks change from having a lot of uranium to less uranium and more lead. So that just means that what we can do is take the rock, look and see how much uranium there is compared to the amount of lead. And obviously, the less uranium and the more lead there is, the older the rock. So that's basically how we use the radioactive dating in rocks. It's just that uranium converts into lead as it undergoes this decay. And therefore, the more lead there is compared to uranium, then the older the rock actually is. Okay? Right, can you go through all the colours for chemistry again? Okay, so colours for chemistry. Starting off with the flame tests, which are looking at the alkali metals then. So you've got lithium, which is the red one. You've got sodium, which is orange, and potassium, which is lilac. So we've got those three colours for the flame test first of all, okay? The next one that we've got then, the halogens, these are obviously just the colours that they exist in. So you've got a green gas, which is our chlorine, you've got bromine, which is an orange liquid, and then you've got iodine, which is a grey solid. The next set of colours, which we haven't mentioned yet tonight, are the transition metals. Now, the transition metals then, you've got copper, which is a blue colour. You've got your iron 2, so the Fe2+, and that's the light green. And then you've got your iron 3, the Fe3+, which is orange-brown in colour. When we carry out the precipitation reactions with them, so this is just looking at copper, iron 2, and iron 3, what we're doing is adding the sodium hydroxide to them. So once you add sodium hydroxide to this chemical, if we get a blue precipitate, so a little blue solid in the tube, then that tells us we have copper ions. If it goes a greyish green colour, then that's going to be the iron 2. And if it goes orange-brown, that's iron 3. And the last one is obviously looking at testing for sulfates. So we've got the white precipitate if sulfates are present, and then the testing for the halides with our silver nitrate solution. Then chloride ions make a white precipitate, bromide ions make a cream precipitate, and iodide ions make a yellow precipitate. So that's the full range of our colours there. The ones that I would say are quite likely to come up, obviously the testing water ones with the halides and the sulfates, the precipitation reaction ones, okay, make sure you get those colours the right way around, particularly for the two types of iron, and the flame tests. I would say they're probably the more likely ones to come up, but they do like that table with the halogens and predicting properties and things like this. So anything else other than the bits I've already gone through, folks? So any other questions, ping them up in the chat window now, and I'll see if I can go through any more for you tonight. There's 10 of you there, and you're all sitting very quietly, which is unlike you all, really. So anyone got any other questions tonight, or are we all now just sitting there contemplating if there's anything else we would like to ask? I'll just give you a couple more minutes, and if we haven't got any more questions, then we'll call it a night tonight. So you can all go to bed, and then I can as well, which would always be nice. What is active transport? Active transport is just the process by which we're going to be able to take up the minerals from the soil. So it's basically when we've got to move something from an area of low concentration to an area of high concentration. So what we find is in the soil, then all the minerals are actually in a low concentration in comparison to the inside of the plant. So we're going against the concentration gradient there. And in order to actually move those minerals from the soil inside the plant, we need to use energy and these special proteins in the membranes to do that. So what you end up with are, you've got these special proteins within the membrane, and then the low concentration of the minerals on the outside, the high concentration on this side, so that means that we've got to actively pump them across the membrane using energy in order to get them inside. Mrs. Buchan's going to be in room 35 tomorrow, Joe. So one more minute to get any of your other questions in, and then we're going to call it a night on your Q&A session as well. for the tracers, if you were asked to pick the best one to use, what features would you consider? In terms of the tracers, if we're looking at ones used in the body, then they need a short half-life, okay? Because we don't want any radioactive substance persisting in the body for a long period of time. But they also need to be penetrating enough to obviously pass from inside our bodies to outside. So that's why alpha is never a good choice for a tracer. If you're thinking about tracers used in industry and things like gas mains and so on, then what you need to consider there is a longer half-life, because otherwise it will have obviously degraded to the point we can't detect it before it gets to the other end. And also you need to make sure that it is one of the more penetrating ones, because remember, those pipes are going to be buried quite deep underground, so it's got to get from the pipe to obviously the surface all the way up here. So they need to be more penetrating and a slightly longer half-life than the ones we use in medical tracers, just because they've got further to go. So hopefully that answers your question there, Ron. Okay, so all we need to understand there is just think about what it's going to need in order to actually kind of make it useful to us. So it's a case of what does it have to travel through, and that obviously links into the penetrating ability, and then follow that up with how long should it last. And if we're thinking about it being inside people, shorter is better, because obviously exposing yourself to radioactivity for a longer period of time is a bad plan, okay? But when you're thinking industry, it should be longer, because otherwise it will have degraded to the point we can't detect it when it's out actually traveling through all those pipelines. When you're thinking about an ecosystem, all we're talking about there are all of the living things interacting within that given environment. So you've got the living things, you've got the different environmental conditions, etc. All of those put together make up the ecosystem. Has anyone else got any other questions? As soon as my clock hits 10 past, then I am going to call it a night tonight, folks. So last questions coming through now. Get them typed quick. A circuit breaker is basically, that's the thing you've got in your house with all those little switches and stuff like this, so that if you get a surge in current that could obviously cause harm to either the devices or the people inside, then it just flicks a little switch. So that then obviously turns off that circuit until you decide to fix obviously the fault first of all, and then you can just flick the switch back. So it's basically just a safety device that we use in our homes in order to then break the circuit if there is a fault. So sedimentation, filtration and chlorination. Filtration, first of all, so that's where we're going to remove all of these larger particles. So any sort of like twigs, leaves, stuff like this. But as it goes through the filter, they get removed. So that then leaves us with very small particles like clay and things like that. And then we allow those to sink to the bottom in sedimentation. And then finally, the chlorination stage is where we're going to add chlorine gas to the water, and that kills the bacteria as a response, therefore making it safe to drink. Okay then, folks, so hopefully I've managed to answer all of your questions then. Obviously, if not, then pop along and see your science teachers in the morning before your exam. Remember to get some sleep tonight, because the last thing you want to do is stay up all night stressing, because at the end of the day, you've done a lot of work and there really is no need to stress about it. So all of you that obviously go to my school, I will see you at some point tomorrow, I'm sure. And for those of you that don't go to my school, then also good luck in your exams and have a nice evening, everyone.